In this video, we will discuss about these T cell activation results post mKRAS vaccination. We have discussed about what they called as mKRAS vaccinations. So in the previous video, what they did is, in the previous video, we have seen the results where they treat these PBMCs from the patients who are affected with the pancreatic cancers with these different peptides of this mKRAS. And in the previous video, I did not said that they expose this mKRAS peptide to these PBMCs for 24 hours before they check the T cells activations against this mutated KRAS. So this is what one of the clarifications that I want to give you before I describe the results that are presented in this figure. So now let us go through the results that are presented in this figure. In the panel, what they showed is that they check the T cell activations through this IFN-Y ELISPOT. So basically, once the T cells gets activated, this is one of the markers that it releases, that is IFN-Y. So they check the IFN-Y levels pre-vaccination and post-vaccinations PBMCs, and then compare their expression levels of the IFN-Y. So what they are able to find out of the 12 patients that they have tested, one of them is immune non-responder, that one of them does not show T cell activations, whereas 11 of them shows the high increased or elevated level of IFN-Y. So they call them as immune responder. And so this is how they define this immune responder and non-immune responder. Exact criteria, they did not mention it in the main figure. Maybe they might have mentioned it in the supplemental figures. I have not gone through that supplemental figures in detail. So out of this categorizations between immune responder and non-immune responder, they checked it against the individual antigens, against the individual mutated KRAS antigens. It is G12V, G12A, G12R, G12C, G12D, and G13D, and they were able to find out that against these 6 mutant, most of this patients PBMCs shows elevated level of IFN-Y. And when they check the quantifications, they are able to see that in terms of the statistics, 53% shows a elevated level of IFN-Y compared to the baseline. And in terms of the fold change, only for the 2 patients that has these G12D mutations are been found to be non-responder, whereas all of them shows elevated levels of this IFN-Y, and especially in the panel D, we can see that for these G12R mutations and G12V mutations, many patients shows elevated or high fold change compared to the case where patients have these G12D mutations. And similar results they have seen when they did the time dependent studies, that G12V and this G12R IFN-Y productions is outperformed the other mutations, that is G12D mutations. And they have also seen that this G12D mutations levels lying with this G12C or aligning with this G12C signals in terms of the IFN-Y levels. So this last information is not that useful at this point of time, but one information that they come out is that the patient with G12R and G12V are more responsive towards this mKRAS vaccinations compared to the case where patients have these G12D KRAS mutations. So this shows the specificity of this kind of vaccine, that it will perform better on the patients who have this G12R and G12V mutations in the KRAS. However, this is not the final point. This is just the second main figure of this paper. We will discuss more results in our upcoming videos, and how they come up to a point, and what are the limitations, and what are the eligibility criteria in order to use these mKRAS vaccinations, along with the two other drugs that we have discussed in the previous video. But we have not discussed the functions of other two drugs in the previous video that we will cover in subsequent videos. So that's all for this video. If you need to understand more about these experiments and their results in detail, and also wanted to look into the supplemental figures, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.